Okay, we've got just two more sections in this chapter. We're going to cover them quickly. This section here will be a lot of review, and then the next section is just some general information that we'll build on in the next chapter. So, stereochemistry. So, if we look at stereochemistry, hopefully you remember from Gen Chem when we talk about that, here are two different compounds, R-carbone and S-carbone, and they only differ at the configuration at this point here. They have identical melting points, identical boiling points, but they actually smell different. One smells like spearmint and the other one like caraway seed. And so if you have chiral molecules, those that have four different groups around a central carbon, the only way that you can distinguish between them is by the way that they rotate plane polarized light. So just as a brief introduction into that. So if you take light, it actually will put out waves in all different directions. Ordinary light, the waves are emanating in all different directions. If you have a polarizer, essentially it's a fine grating that only allows light of one orientation to go through. And so if you were on this other side here and you had a grating as well, you could see the light if your grating aligned with it. If you happen to turn your grating a little bit, it would actually turn dark. So if you happen to have polarized lenses, this is kind of a fun thing to do. You hold it up to a light source and you turn those lenses and they'll go dark. And when you align them, then you can see the light come through. So if you have a sample of something in solution, you can actually pass this plane polarized light through. Now, if the compound is not what we refer to as optically active, and there's a couple ways to be non-optically active, that's either you're not a chiral molecule or you have equal mixtures of a chiral molecule known as a racemic mixture. If you happen to pass plane polarized light through there, then when it exits, that light is passing in the same direction. Those waves are in the same line there. Now, if I happen to rotate that plane polarized light, then what we would say is a molecule is optically active. So here's just another view of it. So we're sending the light through, it's plane polarized. We send it through a sample, and that sample interacts, or sorry, the light interacts with that sample and causes the light to rotate. And so then when the light is coming out, if I had my reader this direction, I wouldn't be able to see any light at all because the light needs to pass through this direction. So what you do is you rotate it till those fringes line up with that light. Let me do a little bit better. So I need to rotate it so the diffraction would line up that way, and then this light would come out, and I'd figure out how far I rotated that, either plus or minus, and that would allow me to determine what we refer to as the optical rotation. So here's just another example of it where we've actually turned the analyzer and you can actually see that plane polarized light come out, and you can actually determine which way the light rotated. And so they refer to it, if you rotate to the right, it's dextrorotatory, and left, level rotatory. Again, none of this should be new to you. So they found out that chiral samples did this. And then what they discovered is that along with this, as I mentioned, these compounds right here are mirror images of each other, what we refer to as enantiomers. And so the melting points would be identical, the boiling points would be identical, but what is different is the way they rotate plane polarized light. Now it's equal, but opposite sign. And so this is a new physical property that we can give to chiral molecules. And what you can see down here is, again, it's equal in magnitude, but opposite in sign. And so you could actually go through, and if you had a pure sample of cholesterol, you could see that, take its optical rotation and see if you got the negative 31.5 degrees, and it would be pure. You know, you could test your cocaine and make sure that it's pure cocaine, or if they cut it with something else, it would have a different. If it was positive 16, then you have the enantiomer of cocaine. And to be honest, I'm not sure what that does to you. So some of you caught on in one lecture that I gave a bonus point. So I'll give a bonus point here. So if you take the enantiomer of cocaine, does it have any physiological activity? So email me and we'll give you a bonus point for listening and figuring out if the enantiomer of cocaine has any physiological properties. Okay, but we want to talk about amino acids, but we first need to understand where this term L amino acid came from. So I mentioned that briefly, that all natural amino acids are L, and it comes from L-glyceraldehyde, which is actually the simplest sugar. So we'll talk about that. But in 1891, Emil Fischer proposed that stereoisomers of glyceraldehyde can be designated as D or L. So if you drew them in this form here, so the highest oxidized group is at the top, then this OH would be off to the left. Now, if you do the same thing here, this highest oxidized group here, then the amine group ends up being off to the left. So these are referred to as the wedge and dash drawing, but we can simplify them and just draw these as lines with the understanding that everything that is horizontal is pointed out at you, and everything that is vertical is pointed away from you. So the Fischer projection allows us to look at a molecule. They're somewhat cumbersome in some ways and somewhat nice in other ways. So if we actually look here, we can actually see that this is what would be referred to as a D configuration there, because that group is pointed off to the right there. So we use these Fischer projections, but there's some things that we need to be able to do, and that is be able to figure out if there are an S. Now, typically when you have a Fischer projection, they put the lowest priority here on the horizontal, excuse me, just the way that it works out. And so what we do is we count from 1, 2 to 3, but remember the lowest priority is pointed out at you. So this looks like it's R, but since the lowest priority is horizontal, it's pointed out at you, it really is S. And then this one's 1, 2, 3. Okay, I've got to count from 1, 2 to 3. I ignore the 4 other than it's pointed out at me. So this kind of looks like it's S, but it's actually the R configuration. Okay, like I said, in the Fischer projections, it just happens quite frequently. The lowest group is out at you. So as we count 1, 2, 3, again, that kind of looks R, but remember the lowest priority is pointed out at you. So it's S, and this one is R right there. Now, there really isn't a connection between D and L, plus and minus, and R and S. Okay, D and L comes from the way things appear in the Fischer projection. And when we draw something in the Fischer projection, just imagine if I change this group right here to a BR. That's going to become a higher priority, even though it still may be, actually, let's do it up here. Now, they typically look at the OH in this. We'll talk about that in carbohydrates. But if I put a BR there, I'm now actually going to change my prioritization around there. So even though we still might consider this a D sugar because the OH is out to the right, we would actually change our configuration. So I don't want to stress that too much other than DL is different than RS, which is different than plus minus. Plus and minus is an experimental determined. R and S have their different set of rules, the Kong-Ingold-Prelog rules, and D and L have to do with how you draw them in a Fischer projection. So they're not always equal. In fact, here's one example here. What we've got off to the left there is actually a sugar in the L configuration. You can see that OH is off to the L. These aren't absolute Fisher projections, because Fisher projections would just have lines, but this would be the L configuration, that's the L configuration, and that's the L configuration. Now, if we determine if it's RS, you can see that this is S there, this is S there, but this is R. And the reason it's R is because the thiol has a higher priority than the OH. In fact, 19 of the amino acids are chiral, all except glycine. And of those 19, 18 are of S configuration. It's only cysteine that is of R configuration. And then don't forget that there's 2 amino acids that have additional configurations, threonine and isoleucine. Okay, so I just want to make sure that you don't confuse that DL, RS, and plus minus are all the same thing. They all are classifying the molecules by different rules, and plus and minus by experimentally determined data. So why do we care about stereochemistry? Well, our bodies are chiral. We're going to find out that proteins tend to contain L-amino acids. We actually know that, but these proteins are actually going to create fixed 3-dimensional space. So if you imagine that this was some type of receptor in your body, you can see that this group matches up with the circle and the square and the triangle. And so that would fit in there. Where if I had the different enantiomer, it wouldn't fit in there. So to kind of put that into perspective, here's a drug known as thalidomide, first synthesized in 1953. It was sold as a sedative, hypnotic, anti-nausea, and leprosy. Well, people that tend to get nauseous are women in their first trimester of pregnancy. And so they gave this to them, and the women were happy. They no longer had morning sickness. Well, the problem is there was some unintended side effects, and that unintended side effects ended up being thalidomide was a tetragenic compound, and it ended up causing birth defects where the limbs and digits were shortened. Otherwise, the children were mentally as capable as any other children. They just had these physical deformities. And so you can see that one of them was an anti-nausea drug and one was a tetragen. Now the problem in our body, even if you gave this enantiomer, your body would convert it into that. So it was a drug that helped with nausea, but had some very unintended consequences. And like I said, one of the reasons is because the receptors in the body were different. And so we actually have some drugs that we take. So ibuprofen, when you get it, only one of the enantiomers is actually an anti-inflammatory. In fact, when you buy it in the US, it's a mixture of the two. And the other one actually makes it a little bit slower for the onset. So you get a 500 milligram pill. Well, no, they're 200 milligrams. A 200 milligram pill here. But if you go over to England, they sell the pure enantiomer, and it's like in 93 milligrams. So they actually can give you less of a single enantiomer, and it actually has a faster onset. Here in the US, they just can't make money selling you the single enantiomer, so they don't. Fluoxetine, also known as Prozac, is a chiral molecule, and naproxen, sodium, this one they have to sell you as the pure S because the R enantiomer is a liver toxin. For ibuprofen, the other enantiomer doesn't do you any harm, and same with Prozac. It doesn't really do you any harm. It causes your liver to work a little bit harder. But anyway, so chirality is very important in drugs, and the bigger picture here is why these drugs have different effects is because they interact with proteins that are also chiral, and they interact differently with them. So I started off talking about stereochemistry, and we talked about carbone and S-carbone, and they smell different. And they smell different because they have they're different enantiomers. So they smell like caraway seeds, which I don't find is necessarily a pleasant smell, but kind of a dill type smell, and then R-carvone smells like spearmints. And I have both of them in my office. If you ever stop by, ask and I can let you smell. And you can see that you can tell the difference. Your nose actually can distinguish between those because you have these olfactory receptors in there. And what I say is it's like little gloves up your nose. So let's say that you have a right-handed glove and a left-handed glove. And so if the molecule fits into that right-handed glove, then it smells like R-carvone or spearmint. If it fits into the left-handed glove, then it smells like S-carvone or caraway seeds. So our body is actually set up to recognize this chirality. And again, amino acids are chiral, and we need to understand that. So there are some things that you should be able to do. And finally, this last little section, there are more than just the 20 amino acids in our bodies. Some of them are modified after they're included in the protein. So you can put phosphates on them. You can add additional carboxylates. So you can see these things in blue are added. You can add an OH group to proline. You can add a methyl group to histidine. You can add an acetyl group to lysine. So there's lots of ways that we can then go add additional complexity into our proteins after we synthesize them. But typically, there's only 20 amino acids. And with that, I have to kind of correct that because we have what are called the 20 canonical amino acids that are included directly into proteins, but we found that these two down here, seleноcysteine and pyrrolysine, in some organisms are actually incorporated into their proteins at the ribosomal level. All these other examples here, these are what we refer to as post-translational modifications, where we go and modify after the protein has come off the ribosome. So just understand that we can get additional complexity in our proteins by modifying the side chains. And some organisms actually have additional amino acids. So let's just talk about some of the things that we can do. There's a little box in there that talks about the green fluorescent protein. This was the previous bonus assignment. I couldn't remember what it was. It was a jellyfish. 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 And I should have remembered jellyfish. So anyway, it came from a jellyfish. Now, like I mentioned, these aren't bioluminescent. They don't create light, but if you shine a black light on them, if you take it to a rave, take a jellyfish to a rave, it will be the coolest thing on the floor because it will fluoresce. And what it does is it takes a sequence of serine, tyrosine, and glycine, and it does this little modification of the backbone, and you get conjugation through this system here. Now that conjugation isn't enough to make it colored, but it is enough to allow it to fluoresce. And we've taken that protein and we've included it in many different organisms. It folds up into this beta barrel, which we'll talk about in the next chapter. And you put the tyrosine right here in the middle, you've got this modification on the backbone there, and they end up being fluorescent. And since it's a protein, if you put that code in the DNA, you can eventually get that protein to be expressed in the organisms. And so you can actually make these, we call them glow in the dark, but they're really not glow in the dark. They just fluoresce in the dark. So like I said, at a rave where you got lots of black lights, these guys would be pretty cool. And so you can actually modify this to get different colors to fluoresce. Here's actually some E. coli that are fluorescing. So that protein has been included in a plasmid, and then that plasmid, that information's been taken from the DNA and converted into a protein. And so they do this because then they can tell what is being made in a cell. So it's actually a really useful tag for lots of interesting things. Go look up the Brainbow. So they've included multiple different fluorescent proteins in brain tissues, and so you can kind of see what is being turned on and off at different times. So look up Brainbow. So this did result in a Nobel Prize, and they've expanded it out where you've actually got many different fluorescent proteins. And they use these again as tags So they append it to different proteins so they can tell when certain things are turned off and on. So I believe this is a C. elegans, a little worm, and I'm not sure what they're detecting, but wherever it's fluorescent, that is attached to some other protein of interest. So let's just say that those are nerve cells, and now you can see where the nerve cells are located in this little worm, or it could be some other protein of interest. So you just attach it. It tends to be small, often doesn't interfere with the natural protein, and it just shows up in the same localized region and fluoresces. And so, yeah, go check out Brainbow. Now, as I mentioned, a lot of times the side chain is modified, but there are two amino acids. They're uncommon, but they're encoded in DNA and inserted during translation, meaning on the ribosome. And then amino acids actually have other roles in cells besides being incorporated into proteins. We'll talk about that in a second, but just wanted to mention these two interesting amino acids. These came about in my lifetime, so as you get older, there's more information to know. I used to only have to know 20, but now there's actually 22. So the 21st, selenocysteine, it's incorporated at what they refer to as a stop codon, which we really haven't talked about in DNA, but it doesn't really have its own codon. It just kind of slips in. And what was I going to say? Yeah, so you can actually get, since selenium's actually just beneath sulfur, you can actually get these diselenides, so selenium bonds, and they're more stable than disulfides. So you can actually have these selenium bonds, and they have special redox roles. And so in 2003, they identified 25 human proteins with selenium. Selenium is an essential mineral. You need it in trace amounts. If you don't get selenium, leads to some problems. And here's pyrrolysine. So it's found in a few species of bacterium. So anyway, so these aren't very common, so we'll just kind of focus on the 20, but there are some organisms out there that are rewriting the genetic code and including other amino acids. But those 20 amino acids are fairly consistent among all organisms. But as I mentioned, amino acids just aren't for proteins. We do use them. So this compound here, gamma aminobutyric acid, is a decarboxylated glutamate amino acid. It is a neurotransmitter. Histamine right here, if you're suffering from allergies, this is a decarboxylated histidine. And this causes mast cell degranulation and other issues that give rise to the symptoms of allergies. Dopamine is derived from tyrosine, and thyroxine is also derived from tyrosine. So our body actually takes amino acids and actually will make hormones out of them. And so in fact, glutamate is actually a neurotransmitter. So these amino acids, besides being found in proteins, have roles as hormones and neurotransmitters and second messengers. So again, our body's taken something, amino acids, and maximize their usage. Just like we talked back about nucleotides. They're not just found in DNA, they're also found, like ATP, as a energy source. I say that with quotation marks, but we use ATP and a bunch of other different modified nucleotides in our body, and we use these modified amino acids. So our body kind of limits the number of biomolecules we have and then expands by making modifications to those limited number of biomolecules. And then finally, I just wanted to talk one last little bit about glutathione again and answer those questions, see if you can recall what amino acids are. So again, glutathione, very important, involved in a huge number of redox reactions in the cell, found in fairly high concentration. So like I mentioned, oxygen is usually not the electron receptor, but what it does is form this stable disulfide bond. Now, hopefully you're able to catch on that what happened was it actually formed this peptide linkage through the backbone. So this is actually glutamic acid right here. So here's the nitrogen, here's the carboxylic acid, there's the alpha carbon. So it's actually the side chain that we're forming that bond to. The next amino acid is cysteine, and then the last amino acid is just glycine. So an interesting tripeptide where we're actually forming a bond with the carboxylic acid of a side chain there. Okay, we're going to end it there. So that was much shorter than the other one. So know your amino acids, know generally about chirality, and understand how we can modify them to get some other uses out of them. And we'll jump into the next chapter and show you a lot more about proteins. But in order to understand that, we definitely need to know about the amino acids.